So this is part 2 of the chapter 43 lecture, and we left off on the development of these white blood cells. So the pluripotent stem cells are found in the bone marrow, and they differentiate to become either a lymphoid stem cell or a myeloid stem cell. And so the myeloid stem cells are what make up all of the leukocytes that are involved in the first and second lines of defense. So the inflammatory response is going to come from the basophils. We have eosinophils, neutrophils, monocytes, platelets, and erythrocytes, which are our red blood cells, all come from the myeloid stem cells. But the other types of immune system cells, the B cells and the T cells that are part of the adaptive or acquired immune response, which are the lymphocytes, actually come from a different set of stem cells called the lymphoid stem cells. So we're going to look a little more carefully at the different players in this immune response. So for leukocytes, we also call them the phagocytic white blood cells. They are attracted by chemical signals or by certain receptors, or actually ligands found on the different pathogens. So chemical signals can be released by a damaged cell. So a cell that is undergoing attack will send out a chemical signal. They enter the infected tissue, engulf and ingest microbes, and they use lysosomes to do that. And we'll look at that in the next slide. Some of these types of leukocytes, again, are neutrophils, which are the most abundant white blood cell. They make up about 70% of our white blood cell count, and they only have about a 3-day lifespan, so the body has to make a lot of these on a constant basis to circulate throughout the body. Macrophages are larger cells. They are the big eaters. They live longer than neutrophils. And natural killer cells are actually cells that destroy any natural or self cells that have become infected by viruses or that are not behaving normally, like in cancer cells. So natural killer cells are responsible for the apoptosis or destruction of an infected cell. So this is some pictures. These are some pictures of phagocytes. You can see here a macrophage that's attacking a fungus, a yeast down below. And you can see here how a macrophage will use its lysozymes to fuse with a vacuole containing a pathogen. So pathogens will be engulfed by the macrophage. So we have endocytosis occurring here, and a vacuole is formed around the pathogen or the microbe. In this case, it means it's a living pathogen. And then we see the fusing of a lysosome with enzymes, which will then be used to tear apart and break up the microbes, and then they're released back out. So these phagocytic cells can detect different components of a pathogen through receptors that are very similar to the toll receptors that insects have. Every mammal does have what we call toll-like receptors, or TLRs, which bind to fragments of molecules that are characteristic of a specific set of pathogens. So even though this is not a specific immune response, we do have different types of toll-like receptors that are going to identify certain types of pathogens. So this is how the immune system actually determines that something is not self. It is something that is a foreign invader. So these toll-like receptors, for example, they're called TLR3s, TLR4s, TRL5s. They each have a specific type of thing that they're going to recognize. So for example, a toll-like receptor 5 will recognize flagellin, which is a protein that is going to make up a bacteria's flagella. So in that way, it's not going to recognize a specific bacteria, but it will recognize a bacteria's flagella in general and then cause an immune response to get started. In each case, the recognized macromolecule is something that's not normally present in the vertebrate body, and is something that is present in a certain class of pathogens. So once these are detected, the phagocytic cell then knows to engulf them and trap them in a vacuole. And again, the vacuole then fuses with a lysosome, and the pathogens get destroyed within the lysosome. And there are actually two ways. We see here, it looks like we just have enzymes going in here, but there's also a gas that is released by the lysosome that actually poisons the pathogen. Okay, so let's see. On to the next slide. Natural killer cells, again, the way that they work is that they release perforin, which perforates cells. So perforin is a protein that is found within a natural killer cell. And this is the natural killer cell, this larger here, and it has a vesicle containing perforin. And the perforin will then go into the infected cell and cause it to have holes made in its cell membrane. So it basically is going to puncture that cell membrane and in that way lyse the cell. It will cause the cell to rupture. So these are the only of the different leukocytes that do not engulf the either pathogen or the infected cell. They work in a little bit different way by releasing something that's going to kill the cell instead. When a mammal's immune system recognizes a pathogen, it can trigger the production and release of a large number of peptides and proteins that also attack pathogens or cause their reproduction to get impeded. So some of these can function like an antimicrobial peptide that can be found in insects, which damages the pathogens by disrupting their membranes. Then there are other types, one of which is an interferon, which works specifically on virus. So interferons are proteins that are secreted by a body cell that has already been infected by a virus. And what it causes that cell to do is then release this interferon, which will tell the neighboring uninfected cells to produce a substance that will inhibit the virus's ability to reproduce. So in this way, interferon will limit the cell-to-cell spread of a virus and help control a viral infection. Some white blood cells also secrete a different type of interferon that helps activate other macrophages and gets them doing more of their phagocytic work. Interferon can be produced by recombinant DNA technology and actually has been proven effective in the treatment of certain viral infections like hepatitis C. Another type of protein or a set of proteins is what is called the complement system. And these consist of about 20 to 30 proteins that circulate in the blood plasma. And they're inactive as they circulate around. But once they come across the surface of a pathogen, they become activated. And what will happen is a cascade of biochemical reactions that leads to the lysis of the invading cells. So the complement system functions both in inflammation, which would be the second line of defense. It's also used in the acquired immune response, so also in that third line of defense. The complement system is one system that actually works in both the innate and acquired immune systems. So the inflammatory response, this is triggered by some sort of damage to tissue, and it's a very specific response. So one of the chemical signals of the inflammatory response is a chemical called histamine, which is stored in vesicles of what are called mast cells, which is a type of connective tissue. So it's going to be found near the surface in epithelial tissue. Right below the epithelial tissue, we have connective tissue containing these mast cells. And within the mast cells, we have histamine. So when there's an injury or a cut occurs, these mast cells will release their histamine. And histamine works to dilate and increase the permeability of nearby capillaries. So what will happen next is that macrophages will be activated, and other cells are going to discharge cytokines, which will signal molecules to beef up and start the immune response. Cytokines work to increase local blood supply, and they're responsible for the redness and the heat that we associate with an inflammation. So again, when you have a cut and it turns red and it feels warm to the touch, that's the result of the action of the cytokines. What's going to happen is that the capillaries are going to leak fluid into neighboring tissue, and that causes the swelling. Other results of this is going to be enhanced blood flow, and that's going to deliver clotting agents and also antimicrobial proteins to this injured area. So the blood clotting is going to occur as part of the repair process, and that's also going to help block the spread of more pathogens elsewhere. Other cells that are nearby that are uninjured are going to attract neutrophils and macrophages. So there are signals that are going to be sent out by other skin cells to attract the neutrophils and macrophages to the site in case there are any pathogens in this injured area. Finally, when you have a cut, you'll notice an accumulation of what we call pus, which is really a fluid that has a lot of white blood cells, and it's made up of both those white blood cells, the dead pathogens, and different cell debris. So you can see in this picture more clearly how we have something like a splinter entering the epithelial tissue. On that splinter are bacteria. And what's going to happen is a chemical signal is going to cause the blood vessel, this capillary here, to start dilating in order to allow the different portions of the blood to, particularly the white blood cells, the phagocytes, to be released into the tissue area. So sometimes we have a local response that's not enough to deal with an attack. And so when a person's temperature gets elevated, that's actually a systemic response to infection. So activated macrophages release what's called interleukin-1, which triggers the hypothalamus in the brain to readjust the body's thermostat to raise the body's temperature. And the reason for this is that a higher temperature can often inhibit the growth of bacteria, but it also stimulates phagocytosis